Today it's a quite interesting topic, and we are looking at the development of biologics in veterinary medicine, which is, I'm not saying a new topic, but relatively unexplored for the veterinary branch. There was reports that while in human medicine, almost 15% of the candidates are large molecules, in veterinary medicine, we're still at 3%. And so we thought maybe it's good to have a little bit of an overview and see how we can contribute to the work of the community. And today we'll do this together with Dr. Jyothirmai Nethi. She studied for a master's thesis at the Indian University of Technology in Guwahati. I hope I didn't slaughter the name, or probably I did, looking at the membrane binding properties of the LCI peptide on Bacillus subtilis. And then she moved to the University of Heidelberg, where she did her PhD in molecular biology, looking at influenza A infection, and all this before joining Lab for Pulse. So we welcome today Dr. Jyothirmai Nethi, and let me see, she joins.
Hi.
Hi, Nethi. Great to have you here today. Thank you
Thank you
for
for
agreeing
having me.
to this stream today. So let's jump to the topic straight away. We were talking a lot about, like, in the last weeks, about biologics, development biologics. What do you think are the most important things to consider when developing biologics?
I would say for both human and vet therapeutics, the most important things are efficacy and safety. The two main important things that one should consider during the development of drugs.
Okay, and I guess if you talk with the people more in CMC, they will obviously tell you all about manufacturing and what this has to do with the rest. But you said safety and efficacy. How do you assess them? Like, are there already something like methods or whatever that we can take from human medicine, or how is it that it's done at the moment?
Traditionally, the efficacy and safety are measured in vivo by performing in vivo studies, like using animals and testing the drugs in question. And nowadays, there's also more development towards the in vitro studies as well to test this even beforehand, thereby also reducing number of animals in the clinical trials. So yeah, they do both in vivo and in vitro studies, while the in vitro studies are still being developed day by day.
All right, and so obviously reducing the number of animal is something that everyone is trying to achieve. And but not just in human medicine, but I guess also in veterinary medicine, when whenever you can reduce the use of animals in research in general, that's an optimal thing. But I was reading an interesting post a few days ago, where people were saying, hey, like, if we do tests on Beagle, for example, we only have like a 50% overlap compared to human. And funny enough, like, even if I'm doing testing on mice or rats, also there I have 50%, probably even less, of overlap with other animal species. So if I go in dog or whatever. So what are the in vitro assays that are actually used to, let's focus on safety, because this is one of the major concern. At least this is one of the main things that is used for the where animals are used in the development of human drugs. So we can take these as parallel. What are the kind of assays that are used in vitro to access safety?
Yeah, I mean, of course, you're right. There is almost not 100% overlap between the humans or other species that are being used for the studies, and I think it's this is like the main point to consider, that the species selection is one of the important things to consider, and for you to do that by performing in vitro assays on different animal studies can also help you to choose the right animal studies for in vivo testing. And for example, at the moment, there is only in the human context, the in vitro studies are being developed, and it can range from in silico screening to MAPS assay, which stands for MHC associated peptide proteomics, and to validating them in the T cell functional assays. But of course, this is also an emerging field. It's only being performed in the human context, and this also has to be adapted to different species as well. And this can actually provide into narrow down the species that we can use it for the in vivo testing, and to use the right species during the drug development so that it's more applicable for humans and also for the vet therapeutics.
Okay. So you mentioned maps. Can you explain
Yeah.
a bit more? What is it about? What like how this works?
So basically, the immune system does not usually respond to the intact therapeutic proteins. Instead, the proteins are taken up by the antigen presenting cells, and they're processed into the peptides and displayed on the MHC class 2 molecules to the T cells. And this presentation is a key step for the formation of the T cell dependent anti-drug antibodies. And that and production can compromise efficacy or safety, and MAPS can capture this experimentally by identifying the peptides that are actually presented to the T cells, thereby shining more light into what are the sequences that are more immunogenic. And with that, we have more information of how we can change the sequences so that they are not highly immunogenic, and basically will help us to do go no go decisions with the protein of
So
interest to carry it
okay.
forward.
So if I understand correctly, you basically feed your antigen therapeutic or candidate or whatever to antigen presenting cells,
Yeah.
let's say macrophages or dendritic cells or whatever. They expose, like they cut down the
There, yeah, generally you can isolate the monocytes, for example, and then you can treat them in such a way that they turn into immature dendritic cells. Then you can treat with the drug of your choice, a monoclonal antibody, for example. And these cells, they already take up the drug and digest it, and then they already present it onto the surface. And this, you can already elute the complex of the MHC and the peptide, and you can elute your peptide, and following a series of proteomic experiments, you can actually know the sequences of the peptides after digested. And of course, you have the peptides, but then they also have to be tested, where you can do the epitope validation and perform more T cell assays to confirm.
all right. And so at the end, you kind of have like a library of like different peptides that you
Yeah,
eluted from your antigen presenting cells. Okay.
yeah.
And like, are there particular differences between, the human and the veterinary field in this regard?
Yeah, I mean, not in principle, because this is the method that can also be applied for the vet therapeutics as well. But of course, we do not have the reagents or workflows which are optimized for animals. So this is something that we can work on to adapt it more for the animal therapeutics. But in principle, the methodology stays similar. And how the MHC, for example, it's the form of HLA in humans, they normally digest in a similar way, and the presentation is also done in a similar way. So I think the methodology works similarly. It's just the reagents and
Okay.
workflow has to be optimized.
And the MHCs, I guess, they're also different. So you will have probably to,
Yeah, yeah.
like, is there already data on this? Like, if you have, like, I know for humans, you have probably programs, and so you go on PubMed, or there are more specialized um, websites for epitope mapping, or whatever.
Yeah.
But what is the state in veterinary medicine?
In human, you're right. There is already things that are being done in this aspect, but in veterinary case, it's not. It's still emerging. There is not that much data available, and there are also not many companies who are also performing this or offering this service.
All right. So what could be like a logical next step for the community to get together on this and start developing more, safer drugs or safer biologics?
I think for we could also start with this exercise, for example, to try to adapt it to the animals, and this will not only help for it will help for both human therapeutics and also for the veterinary therapeutics. Human therapeutics in the case that it reduces the you can be more specific about the species and then you make a safer choice of animals. For animal therapeutics also, it will lead to a faster discovery and lead optimization and getting drugs faster into the market. So I think
Mm.
at the moment, the veterinary community has to come together to more optimize it to the animals to see if the same methodologies like this can be applied to them.
Like, I mean, here that there's a lot about open science and sharing data and results, especially for this kind of libraries.
Yeah.
Still, curious to see if there will be many more developers coming up in the next months of years with data on this, and we'll be able to share this. If you look
Yeah.
at the human part, um,
Hmm.
I know that there are quite some big consortia between Roche, Novartis, Janssen, and so on and so forth. Um, they at least they started setting some standards in the essays, because obviously if you have one
Hmm.
protocol, it's different from what I might establish. But
Yeah.
then it's also about how many times do I have to repeat the same experiment across companies to just
Hmm.
validate one epitope.
Yeah. Yeah. Exactly.
Is
And
there any kind
also
of,
like
of consensus there?
I think there are also some CROs, for example, who are offering services of immunogenicity, but only for humans. And then they
Okay.
also mention this can be adapted to animals, but of course, they never reveal what exactly they are doing to make it easier for other companies. Since it's still emerging, I think there is also kind of competition and revealing their workflows, for example.
Right, right.
Yeah.
I mean, this is quite is kind of interesting also for the veterinary field. There's a lot of competition is a lot of things work between CROs and companies.
Yeah.
But I'm looking forward to more development also in academic environments also. Yeah.
Yeah, yeah. I think the most trouble is also getting samples from different animals and not knowing what animals to go for and how can they do it because there is nothing in place that they can follow. So I think
Okay.
this is also one of the issues why there is not huge development in this field, I would say.
Yeah, I mean, also there, I guess. Um, we are dealing with completely different quantities of blood of cells that you can get from different animals. So if you characterize the cells um, for one particular study, then you might be able just to use it in a subset of
Yeah.
studies, while in human you might get a much wider or a much larger amount of cells and repeat it over several studies. So this can also be
Mm.
obviously a
Yeah.
limitation.
Yeah. Of course.
Okay, so do you have any, final remarks before we open to a short Q&A?
Um, um, it's just, I
I
think,
think
to
you
help
disconnected.
the.
Can you repeat? I mean, there was a disturbance in the force, so I don't know if.
Yeah. I would say just for the to bring the drugs for the companion animals and farm animals into the market faster, there has to be more funding and more scientific communication between companies and academia and the industry to work together to make it possible, because at the moment all the reagents and workflows are already based in human and it's difficult to adapt for every single thing as you mentioned. So I think this is where the people have to work together to make it more faster, and this would ultimately help for animals and for humans, because the more we know about how it works for our animals, this can long run also help for the human drug development as well.
Okay, yeah,
Yeah.
definitely. Thank you very much for these remarks. Let's see a little bit what we have in the chat for questions. Just let me see. So we have one question. Which conference? Just a moment. Lost for a second. Which conference you're most looking forward to attend this year?
There is one coming up in March in Munich. It's called Analytica.
Okay.
I think this is something as a team from our company that we would like to attend.
Right. There will be
Yeah.
quite some people in Munich there.
Yeah.
All right. Then I see that there's nothing else coming up for the moment. This is fine. Short, nice round. So let's wrap it up
Yeah.
here. Thank you very much, Neti.
Thank
Looking
you.
forward to the coming discussions.
Yeah.
And then I thank you everyone for attending this live stream. Bye to the next one.
Bye.